Welcome once again to Bio 211D Plant Physiology, Unit 5, Session 5, Mass Pressure Flow Hypothesis. And by the close of this session, you should be able to describe the mass flow or pressure flow hypothesis, explain the mechanism of mass flow or pressure flow, and advance evidence to support movement of organic solids. The pressure flow hypothesis, also known as the mass flow hypothesis, is the best supported theory to explain the movement of food through the phloem. It was proposed by Ernst Mach, a German plant physiologist in 1930. A high concentration of organic substance inside cells of the phloem at a source, such as a leaf, creates a diffusion gradient that draws water into the cells. Movement occurs by bulk flow. Phloem sap moves from sugar sources to sugar sinks by means of tubular pressure, also known as hydrostatic pressure. So a sugar source is any part of the plant that is producing or releasing sugar. But during the plant's growth period, usually during the spring, storage organs such as the roots are sugar sources, and the plant's many growing areas are sugar sinks. The movement in phloem is bidirectional, whereas in xylem cells it is unidirectional, that is upward. So flow, according to pressure flow mechanism, water containing sugar in solution flows under pressure through the phloem. It involves the following. A. Glucose is produced by photosynthesis in the mesophyll cells of the green leaves. Some glucose is used within the cells during respiration. The rest of glucose is converted into non-reducing sugar, that is sucrose. B. It has been shown that the sucrose concentration in sieve tubes in leaves is commonly between 10 to 30%, whereas it forms only 0.5% solution in the photosynthesis cells. The sucrose is actively transported to the companion cells of the smallest vein in a leaf. D. The sucrose diffuses through the plasmodesmata to sieve tube elements. As a result, concentration of sucrose increases in the sieve tube cells or elements. The sucrose is actively transported to the sieve elements. Water moves by osmosis from the nearby xylem in the leaf vein. This increases the hydrostatic pressure of the sieve tube elements. Hydrostatic pressure moves the sucrose and other substances in the sieve tube cells and then moves to sink in the storage sinks such as sugar beets root and sugarcane stem. Sucrose is removed into apoplast prior to entering symplast of the sink. G. Water moves out of sieve tube cells by osmosis, lowering hydrostatic pressure. Thus, the pressure gradient is established as a consequence of entry of sugars in sieve elements at the source and removal of sugar, that is sucrose, at the sink. The presence of sieve plates greatly increases the resistance along the pathway and results in the generation and maintenance of substantial pressure gradient in the sieve elements between source and sink. So the sieve element contents are physically pushed along the transportation pathway by bulk flow. The pressure flow theory accounts for the mass flow of molecules within phloem. As the sap is pushed down, the phloem sugar is removed by the cortex of both stem and root and is consumed or converted into starch. Starch is insoluble and exerts no osmotic effect. Consequently, the osmotic pressure of the contents of phloem decreases. Ringing or girdling, that's evidence to support movement of organic solids. So when a tree is girdled, the phloem is removed, but the xylem is left intact. The role of the bark is in sugar movement in plants. Mason and Maskell in 1928 demonstrated that removing a complete ring of bark while leaving the wood xylem intact prevented downward movement of sugars. When a strip of bark was retained between upper and lower stem parts. In B, sugars flowed downwards in direct proportion to the width of the remaining bark. If a tree is girdled or ringed below the level of the majority of leaves, the bark swells just above the cut and sugar accumulates in the swollen tissue. So when a tree is girdled, the phloem is removed, but the xylem is left intact. So in conclusion, sugars are transported in the phloem, but not in the xylem. Therefore, the result of girdling suggests that phloem is the tissue that transports sugars. Radioactive tracer studies with carbon-14 exposing mature leaves to a carbon dioxide that has been synthesized with radioactive carbon-14 labeled after a period of time. Radioactively labeled sugar is soon found moving down the stem into the roots. Use of exudates from aphids. It's difficult to get samples of sap from phloem without injuring the phloem, but this problem is solved by using aphids, small insects that are phloem feeders. The aphid drives its stylet between the epidermal cells into the phloem. If the aphid is anesthetized using ether, its body can be carefully cut away, leaving the stylet. Phloem sap can then be collected and analyzed. Analysis shows the presence of sugar and amino acids. So in review, we have been learning about the mass flow hypothesis, which is also known as the pressure flow hypothesis. So by now we should be able to perform simple experiments to explain this hypothesis.